I would just like to introduce the speakers this evening. So you will have the opportunity to listen to 3 speakers. The first being Professor Julia Horrocks. You can see here. So just a brief bio. So Professor Horrocks earned her PhD in the behavior of green monkeys, and she earned that PhD from the UWI Cayfield campus in 1985. During a postdoctoral year at McGill University in Canada, while resident at Vallès Research Institute in Barbados, she became interested in the biology and conservation of endangered sea turtles. She established the Barbados Sea Turtle Project in 1987, and the Barbados Sea Turtle Project is now located in the Department of Biological and Chemical Sciences at the UWI, and became Wider Caribbean Sea Turtle Conservation Network, known as WIDECAST, country coordinator for Barbados in 1988. The Barbados Sea Turtle Project has been implementing the recommendation of WIDECAST's Sea Turtle Recovery Action Plan for Barbados since it was published by the UNEP Caribbean Environment Program in 1992. Professor Horrocks is a consultant to Caribbean NGOs and governments and has participated in the development of sea turtle recovery action plans for other Caribbean states. She is coordinator of WIDECAST Caribbean Marine Turtle Tagging Center, also located at the UWI, a member of the WIDECAST Scientific Advisory Board, and a member of the World Conservation Union IUCN Marine Turtle Specialist Group. She has authored dozens of books, management and technical reports, peer-reviewed scientific articles, and popular articles. She was the recipient of the Governor General's Environmental Award in 2001 for her work in conservation of biodiversity, especially sea turtles. Professor Horrocks was one of 5 2004 recipients of a Pew Marine Conservation Fellowship, the world's most prestigious award honoring and investing in applied ocean conservation science and outreach. We then have Dr. Henri Vallès on the other side. So Dr. Vallès pursued a BSc in biology at the University of Oviedo, in Spain, and then taught biology for several years at the State University of Haiti before coming to Barbados to pursue an MSc at the UWI in Surmis. During his MSc studies, he became particularly interested in reef fish ecology, and he continued that line of research while pursuing his PhD at McGill University in Canada. Following his PhD, he took a 2-year break and worked as a technical advisor to the Fisheries Department of the Ministry of Agriculture of Haiti in projects aimed at improving artisanal fisheries in Haiti. Dr. Vallès returned to the UWI and Barbados as a postdoctoral fellow for the Future of Reefs in a Changing Environment, FORCE, project, with the task of helping develop indicators for coral reef management, and he subsequently joined the Department of Biological and Chemical Sciences as lecturer in ecology. His research areas are focused on settlement and early post-settlement processes in reef fishes, threats to coral reefs, artisanal tropical fisheries, ecological indicators, biodiversity conservation, parrotfish ecology, and quantitative methods in ecology. Our final speaker is Dr. Darren Browne, see here. So Dr. Browne has always been a scientist, and he remembers conducting experiments on the dining table as a child, using his mother's compact mirrors to make a periscope and making models of the human skeleton out of plasticine. You all remember plasticine? Yeah. No? Yes. The younger people are shaking their heads like they don't remember plasticine. I don't know what they call it. Play-Doh? Play-Doh? Yes, same thing. His love of scientific inquiry crystallized as when he enrolled at the University of the West Indies Cayfield campus and attained both his Bachelor of Science degree with a biology double major and later his PhD in biology. He considers himself to be a citizen of the Caribbean and is interested in exploring and conserving the flora and fauna of the Caribbean region. As passionate as he is about field work, his interests also include technology and database management. After completing his postgraduate certificate in undergraduate teaching and learning, he has also found that he takes after his parents, who are both educators, and he is interested in maximizing student achievement and creating the same passion for biology in his students that drives his own research. His research areas are population genetics, DNA barcoding, biology of invasive species, and conservation of endemic and endangered species. And before I ask you to welcome all three speakers, I will let you know who I am. So I am Professor Avril Williams, and I am the current head of the Department of Biological and Chemical Sciences at the UWI. So without further ado, please welcome all three speakers. So Professor Horrocks will go first, followed by Dr. Browne, and then Dr. Vallès. Let me replace everything the way it was. Yes.
Everybody, glad to see familiar faces and some new faces in the audience. As Avril said, my name is Julia Horrocks, and I'm going to begin this lecture with just a brief introduction to the topic of invasive species, and then I am going to give you an overview of the roles of the UWI ecology staff in assisting the government to address the problems with invasive species in Barbados. And this lecture is part of the offerings of the Faculty of Science and Technology for the 60th anniversary of the Cave Hill campus. I want to start by making the point that this slide has been up on the screen for quite a while now, and you're probably all very familiar with the species that you can see there. But perhaps some of you may not be aware that these species are either considered to be invasive already, or they have the potential to become invasive. So in other words, they're not part of the native fauna of Barbados. So what we're talking about this evening are invasive alien species. Alien, everybody knows what that means. It means they're not native, they're from elsewhere. And invasive has a particular meaning. It's a non-native organism which has adverse impacts on native species, ecosystems, economies, and even on public health. And invasive alien species, or IAS for short, are a significant problem for the biota of islands in particular, and I'm going to come back to that in a moment or two. The other thing that you'll see there is that the deliberate and accidental deliberate introductions of invasive species have gone on since the islands were settled. So, sort of Amerindian times, species were being introduced. Obviously, post-European colonization, there were more introductions, and a lot of them were deliberate. So the mongoose, for example, was introduced for biological control. The monkey was introduced as a pet, so these were deliberate introductions. And there are other species, of course, like giant African snails, the planaria, which Darren will be talking about in a moment or two, which are accidental introductions. So we can divide them into those two categories. And lest you be in any doubt as to the impact of invasive species, just to give you an idea, $27 billion per year is spent on, is the actual cost of invasive species. So the damage that they do and the expense of actually trying to manage the species. And you'll see from this graph, I'm not sure if you can see at the back, but many of these species are insects, and they are vectors of various diseases, like Aedes species of mosquito, but also rodents, who are, very problematic in terms of crop damage, and also in terms of damaging native fauna and flora. So the impacts on native species, this is something that ecologists are really interested in. And it's not just for the sake of the species themselves, because if an invasive species damages native species, then there are knock-on effects, it damages ecosystems, and ultimately it will have a significant negative impact on human activities. So don't think that addressing the problem of IAS is simply about protecting native species. It's not. So I said that invasive species have been particularly problematic for islands, and I'm beginning with this slide because this is one of the most famous of the extinct species, the dodo. And we all know the expression dead as a dodo. It was one of the first animals to go extinct, and it was driven to extinction both by human activity directly in terms of exploitation, but also because humans introduced two species which were invasive in the Mascarene Islands in the Indian Ocean, and these species damaged the habitat. Dodos are ground nesting birds. They're flightless. They've got very small vestigial wings, but they're flightless. And this is true for many island species of birds, which have evolved in the absence of mammalian predators, because mammals find it quite difficult to get to islands. So flightlessness is a characteristic of many birds on islands. And the pigs and the crab-eating macaques are the species which really finished off the dodos, eating their eggs and damaging places where they made their nests. So if we come forward 300 years and we come to our region of the world, you can see just how connected places are nowadays. These are shipping routes between different parts of the wider Caribbean. And these routes represent ways in which invasive species can be introduced into new areas. And so you can see that there is a big challenge for governments in the region to be able to address the impacts of invasive species. I also wanted to mention an example where an island has actually been restored, because it's one of the examples globally, one of the best examples where an island which was destroyed by invasive species has actually been recovered. This is the island Redonda, which is off the coast of, well, about 35 miles off the coast of Antigua, belongs to Antigua and Barbuda. And it is an island which is famed for seabird nesting, and seabirds produce a lot of guano, or bird droppings, which become solidified into phosphate sediments, which are then mined. So they're very valuable. And in the 1800s, miners arrived on the island and they accidentally brought rats with them and deliberately brought goats with them. And the rats and the goats survived on the island long after the miners left and decimated the populations of birds and reptiles that lived on the island. You can see a helicopter there that was used to actually remove the goats from the island. So they were individually bagged and carried onto the helicopter because you couldn't actually approach the island by boat. It's not possible. The sea conditions don't lend themselves to that. But you can see how bare the island looks, devoid of vegetation. Now see how it looks and it's even better now. We've got shrubs and new trees beginning to grow, and the recovery of species endemic to the island, which are only found on the island of Redonda, nowhere else in the world, and also the recovery of important seabird colonies. You can see frigate birds there and boobies. And I wanted to give a little shout out for the ecology major, which is, which is taught at the UWI. This is Shanna Challenger. She's a major from 2016. And it's Shanna who actually led the restoration of Redonda. And you can see there meeting David Attenborough amongst one of the accolades that she received for her work. Okay, so that leads me very nicely into what I wanted to say about the role of ecologists in advising the government on how to manage invasive species. So the working group on biodiversity is a cabinet-appointed group, which you can see a number of ecology staff, some of them here, some of them are retired, recently retired, and they are on the biodiversity working group, along with, I don't think you can see all the people in the image there, but there are a number of ecology graduates, undergraduates and postgraduates who also sit on that committee. And our work is to prevent new introductions, well, provide advice on how to prevent new introductions and to manage existing invasive species. So what do? The first thing that the working group does is it reviews applications for the import and export of species. So there are a lot of exotic pet fanciers on the island, and they want to bring in exotic pets. And so we work very closely with veterinary services. And, there are people who want to bring in exotic plants. So we work also with plant quarantine. And in order to make sure that we're not bringing in any species who are invasive, that have been demonstrated to be invasive elsewhere. We also provide technical advice on national legislation, which impacts invasive species. So the working group provided the technical info for the development of two important pieces of legislation. One is the legislation for CITES, which basically regulates trade in listed species under the CITES Convention, Convention on International Trade in Endangered Fauna and Flora. And also we have been working on the Zoos Act, which is currently in draft form and is looking at registration of zoos and making sure that holding facilities are adequate, and also that when the zoo closes, or if the zoo closes, that the animals are safely disposed of. And thirdly, we provide technical advice on invasive species projects, and there's a very important GEF project which is going on in the Caribbean at the moment. It's a $10 million project, which is working across the Eastern Caribbean. And Barbados has 3 pilot projects under the GEF, the overall GEF project. The first being the Lionfish Assessment and Management Project, which Henri Vallès will be talking about in a moment or two. And secondly, the creation of a biosecure site for the critically endangered endemic leaf-toed gecko. This species was rediscovered in 2011. We thought that it was extinct, but it was located only in coastal areas, so sort of limestone coastal cliffs. We don't know for sure whether it was pushed into those areas by competitors, particularly Hemidactylus mabuya, which is the white gecko that is very common in people's houses. That is an invasive species. Wasn't on the picture at the beginning, but it is a very common invasive species. So what we're doing for Phylodactylus, the leaf-toed gecko, is we're creating a biosecure area. We don't have the luxury of offshore islands like Redonda, where you can actually clear the island of invasive species. So we have to create our own island actually in Barbados. And this is what is happening. With the assistance of Fauna and Flora International, we are creating a 2-hectare biosecure area, which will have special fencing around it to stop species which prey on the leaf-toed gecko, like mongoose and rats, centipedes, and we are going to try to prevent the incursion also of the white gecko. And you may recognize a face there, Connor Blades. He is recent ecology MPhil graduate, and he is now working on this project, sort of spearheading the project for the government. And then finally, we have the rat and mongoose control at a selected hawksbill nesting beach. Here, this work is being done at Bath. Bath is a very important nesting beach. It has a subpopulation of hawksbill turtles, which is different to the populations nesting on the west and the south coasts. So it's important from that perspective. But it suffers greatly from predation by mongooses. About 40% of the clutches are destroyed by mongooses. And so this project is actually looking at whether mongooses and rats can be controlled at the nesting beach. And in 1 year alone, more than 50 mongooses were trapped at Bath, and this has contributed greatly to the overall hatching success of the critically endangered hawksbill turtle. Okay, so that brings me to the next section. My colleagues are going to be talking about the research that they are doing in relation to invasive species in Barbados. Thank you.
All right, good night, everyone, or good evening, everyone. I'm Dr. Darren Browne, and I'll take you through the next section of the talk. Now, in this section, we're going to be focusing on a few emergent threats, specifically to the island of Barbados. I'll focus on a terrestrial threat, so invasive land planarians, or invasive flatworms. And Dr. Henri Vallès will speak about 2 marine threats, so our lionfish and sun coral. So let's get started on invasive land planarians. All right. Now, there have been essentially 2 different waves of invasions of invasive land planarians around the world. There's an old invasion where the flatworms have been coming to the Caribbean and moving into Europe and so on, generally from locations in the Pacific. And then there's a new invasion. And what's happened is with the old invasion, some of these worms, they were limited to hothouses, greenhouses, and so on, because they were moving from tropical locations to more temperate locations. In this case, in the case of the New Guinea flatworm, so this is a worm that's spreading now, and it's spreading quickly. All right? Now, the New Guinea flatworm, you can see pictured here, it's native to New Guinea, and it's a predator of snails, slugs, earthworms, and millipedes. Generally, any soil invertebrate, it likes to eat. Now, one of the things that has contributed to the rapid spread of the New Guinea flatworm is that it's resistant to heat treatment. Now, normally, any soil plant material that's imported to Barbados is treated at about 34 degrees Celsius. And this is enough to kill eggs, organisms that are associated with that material. Now, the New Guinea flatworm can actually resist temperatures up to 43 degrees Celsius. So it's really resilient and it can survive that initial treatment. And so it can sneak in. Now, they reproduce very quickly. So they reach sexual maturity in about 14 days, in about 2 weeks. They produce eggs in cocoons that they bury in soil. But they produce about 9 offspring per cocoon, which doesn't seem like that many. However, they can produce a new cocoon every 10 days. That's sexual reproduction, but they can also reproduce asexually, and this is common for all land planarians. Any segment of a worm that is damaged can grow into a new worm. All right? Okay. So moving on. So I call the New Guinea flatworm the vanguard of the new invasion. So we have the old invasion, the worms have been here for hundreds of years. And the New Guinea flatworm. So we can look at the native range. So actually New Guinea and Northern Australia, that's where these worms are from. And this is where originally they were limited to this range. And this is the current range. So they spread all over the world. So when Professor Horrocks was talking about deliberate introductions, in some cases these worms were deliberately introduced to new locations, primarily to feed on giant African snails, because they do feed on giant African snails. However, the problem with introducing a generalist predator to a new location in order to try and control an invasive species is that generalist predator may become an invasive species itself. It doesn't just feed on the species that you want it to feed on. All right? Now, what's interesting here is the rate at which this worm has spread. So I'm just looking at some of the most recent introductions. In 2014, it was spotted in Europe for the very first time. In 2015, it showed up in both Florida and Puerto Rico. 2020, identified in both Martinique and Barbados. And then 2022, just last year, it's been identified in Jamaica. So it's spreading throughout the Caribbean now. All right, so why do we need to be concerned about the New Guinea flatworm? What are the threats that this species poses? So they're predators of land snails, slugs, and earthworms. Snails and slugs, generally persons who are gardening, who are farmers, they don't mind something that controls those. But New Guinea flatworms feed on earthworms. And this is a major issue. By reducing the population of soil detritivores, they can actually change the soil structure, they can change nutrient availability to plants, they can change the porosity of the soil and make it more likely to flood. All right, this is a problem that we would be very concerned about in Barbados. The end result of this is potentially a reduction in agricultural productivity. They also are toxic if ingested. Most land planarians actually do produce some form of toxin. They generally, pets, livestock, they're not accustomed to seeing these organisms. So if they feed on them, they're not necessarily lethal. However, they may induce vomiting in pets, in livestock, and so on, and in human beings as well. So if somebody has a baby that picks up something and puts it into their mouth, can be a big problem. They also act as hosts of the zoonotic parasite Angiostrongylis cantonensis. Now, we are not sure at the moment that this parasite actually is in Barbados. This is a parasite that can cause meningitis in humans, and it can be carried by a number of different things, including giant African snails, incidentally. But we don't want to have a new vector of this on the island. So if we do find that it's present, we now know, there's more ways in which people can become infected. All right? Okay, so let's now follow the invasion of Barbados by the New Guinea flatworm. So it's first reported in a residential garden in 2020, in October of 2020, after about a week or so of heavy rain. Someone found these strange, as they call them, black earthworms in their garden. And so we did an initial survey of this site as well as the surrounding area and found about 60 individual worms. Now, whether they were reproducing or becoming damaged with the damaged parts growing into new worms, we weren't sure. But we suspect that they are reproducing now. All right, so this prompted a few different approaches to surveys. We initially did site visits and surveys. So we visited residential homes and gardens. Anyone who would give us the permission to survey their property, we took that opportunity and we looked on the rocks, on the stepping stones, all of these different things. We also surveyed different plant nurseries. And you can see here some of the results. Actually, the student who did that initial survey is right here at the back. Sorry to point you over, Nisha. All right. So we see here, so this is a worm that we found at the plant nursery. This is one that's been injured. So the other piece of that worm is somewhere growing into a new worm. And here is an egg cocoon with its parent right here. Right. So we did find quite a few worms. We did find quite a few sites. Initially, we only had about 11 locations where the worms were found. Now, in addition to that initial survey, I started a citizen science project. So some of you might have actually seen this image. It was distributed with the help of the Ministry of Environment and past students sent all over social media, just trying to solicit reports about locations of the worms. And with that, we've gotten now about 23 sites, 23 reports, 20 different sites. Now, the objective of all of this was to estimate the distribution of the worms around Barbados, but also to compare the conditions, the different environmental conditions at sites where the worms were present with the conditions at sites where they were absent. Right? The eventual goal of this is to be able to predict where they may be able to spread on the island. Right? So let's look here. This is the current distribution on Barbados. And so these are our different reports. We can see most of our reports have come from Christchurch. There's a few from other parishes. So 2 from St. Michael, 1 in St. James, 1 in St. Andrew, 1 in St. Philip. And so when we turn that into a heat map, just looking at the prevalence of the reports, we notice that there's 2 major hotspots in Christchurch. Right? And then these other smaller areas that correspond to just one report, 1 or 2 reports. No, all of this information was combined with data on environmental conditions, population density, and so on, into a habitat suitability model. And the idea of this model is to predict, based on the conditions where the worms have been found, or where a particular species has been found, predict where it can potentially be found in future. The most likely suitable habitat for that species. All right? And we see here the habitat suitability model. This is, it's a false color applied to this. So we're seeing that where we have our blues, it's just in correspondence with temperature. So our blues mean lower probability, and as we increase our greens 50%, and then our bright deep red is over 95% probability that the habitat is suitable. So it doesn't mean that we're 95% positive that the species is going to be found there, but we are 95% positive that it is suitable habitat. So if there's an introduction event, it could potentially survive there very easily. All right, and so looking at this, we see that some of these, the predicted sites, they correspond with the two hotspots, the two existing hotspots in Christchurch. But in addition to this, we have areas in St. Michael, an area in St. James, and we can see both St. Thomas and St. George have areas of over 95% probability of suitable habitat. Now, these graphs here, so remember there were a number of different factors that were considered in making this prediction. And the three factors that seemed to have the greatest influence were human population density, vegetation quality, and temperature range. So the population density, and this human population density in number of persons per square kilometer, you can see that there's actually an ideal level in terms of population density. So just below 1,000 human beings per square kilometer is ideal for establishment of these worms. When we look at the vegetation quality, this is measured by the normalized difference vegetation index, and essentially the lower the value of NDVI, the closer you are to bare soil, and then the higher the value, the closer you are to a completely covered canopy. And so the peak area right here is just below 0.4, and that's sparse canopy. So maybe grass, but some trees, providing shade, but not complete shade. All right? And then the final factor that we're looking at here, this is the annual temperature range. So it's the difference between the highest temperature and lowest temperature at a given site. And so we can see here between a variation of between 8 and about 8.4 degrees. So over the course of a year, sites that vary by about 8 to 8.4 degrees have a higher probability of hosting these worms. Right? So this is a preliminary habitat suitability model, and hopefully as we get more information, we can flesh this out. But hopefully, we can begin with those predictions of where the worms are may show up next. We can begin looking at ways to control the spread. Right. So in summary, for the New Guinea flatworm, so it's identified in Barbados in 2020, currently well established in Christchurch, but has been seen in St. James, St. Michael, St. Philip, and St. Andrew. It may have an impact on soil health and agricultural production, and it may also pose a health risk because in part because if it's ingested, it can cause nausea at the very least, but it may also carry a parasite that causes meningitis. And based on the model, St. Thomas and St. George may be next on its list.
No, please.
All right. Okay, so this ends my section of the presentation, and I'll hand over to my colleague.
Good evening. Can you hear me? Okay, so I'm gonna actually take you from the land to the sea, as you would have probably figured out from looking at that slide. And I'm gonna spend the last 15-20 minutes talking about two stories, or presenting to you two stories that would sort of illustrate the type of work that we do in the water, or more specifically underwater, in this particular case. So I'm sure everybody here is familiar with lionfish. I'm sure at this stage you've either run into it at some point, or heard about it, or actually tasted it if you had the opportunity. Just to give you a very brief historical perspective, lionfish are actually native to the Indo-Pacific. And they're beautiful fish that have been used in the ornamental industry for a long time, so very often used in aquaria. And the story goes that in the mid-1980s, in Florida, perhaps through an accident, some lionfish got released from an aquarium and got into the water. And they actually appreciated very much Floridian waters because they started to reproduce and the population started to expand. And as you can see from the map here, I'm not sure what this was. Okay, it doesn't work. pointer. You can see it first from Florida, it started going upward to the western coast of the United States until a point where it got too cold, and then they decided to turn around and go all the way down to the south. And in a period of 20 years, basically, they took over the entire Caribbean region. This is a species that is carnivorous, actually very unusually looking, as you know. Most of our native fish here in the Caribbean are not familiar with lionfish, and therefore they fall as easy prey to this predator. So, as you can see from this map, I'm not sure it shows clearly, this is 1985, you can see a red dot in Florida, and then by 2011, over a 20-year period, they arrived to Barbados. And this means that we actually, being further south, east in the Caribbean, we could see it, that it was coming towards our way. It was basically inevitable, so it was just, its arrival was imminent. So we work with the government of Barbados to, first of all, raise public awareness about the arrival of lionfish. And in fact, we kept our eyes open, and at the end of the 2011 year, we had the first record of a presence of lionfish in Barbados. And then by the end of, as you can see here from the chart, by the end of 2012, we could see lionfish records being increasing dramatically. And from this map, I'm not sure you can appreciate it, but by the end of 2012, lionfish have been reported pretty much on the east, south, and west coast of Barbados. It had fully become established in Barbados. So why should we worry about lionfish? Well, the data that were coming from other locations in the Caribbean spoke about huge or high densities of lionfish in those locations where it was found. And along with those high densities, some of the experimental evidence showed that it actually ate pretty much it came across. And that, so this is from one of the highly cited paper from the Bahamas, where lionfish reach very high densities. And as you can see here, basically we have reefs where lionfish are present versus reefs where lionfish are absent. And basically what we have here on the y-axis is the abundance of reef fish. And you could see that the presence of lionfish actually decimated the abundance of reef fish. So this caused great concern. And this was a period of quite active research and monitoring. A manual on how to deal with the invasive was published. You would go to any conference in the region, lionfish was, one of the main stories. So, because we saw it coming our way, we decided that it was important to establish baselines to assess the extent to which things would change when lionfish would arrive. In other words, we were trying to measure the impact that lionfish would have on both coral reef fish communities of Barbados, as well as the fisher livelihoods, because obviously if the lionfish are eating all the fish, then the fishers will not have much to fish. And the picture that was being painted in the region was that one of a catastrophe heading our way. So what we did basically, in 2012, just before lionfish became established, we did a number of reef fish surveys across reefs of Barbados, 10 reefs that are representative of the types of reefs that we have in Barbados: bank reefs, patch reefs, fringing reefs. And we sought to characterize the reef fish communities that were there at that time, before the lionfish became established. And at the same time, we also visited, we did fisher surveys at Pile Bay and Oysters, which are two of the primary landing sites for reef fishers. And we surveyed the catch of reef fishers, we looked at composition of the catch, how much they were catching, just to get that baseline information. And the idea was to repeat these surveys once lionfish became established. And this is what we did 10 years after. So we just finished a round of surveys last year where we basically repeated everything that we did in 2012. But obviously, now lionfish was already present in Barbados. So just an illustration of a reef survey, and basically using a belt transect, we have a line in the water of a fixed distance, and then we go along it and count how many lionfish we see, and then also count how many other fish that are potentially prey for lionfish, like parrotfish or surgeonfish, and we size them, and we estimate how much of those different groups of fish we can find on the reefs. So we did that across 10 different sites. Again, in 2012, before the lionfish became established, and then 10 years after. Same sites, same methodology. And we did the same thing for the fisher surveys. So what do we find? Well, the results are somewhat complex, but the take-home message is pretty straightforward. We found basically no evidence of a change in reef fish communities. So what I'm showing in these slides, so we have here the parrotfish, and we have in the green box is the pre-invasion levels. This is biomass, and I would like to for you to focus on the horizontal line here, which is the median value. And as you can see, when you compare it to the parrotfish in the post-invasion period, overall there is not much of a change. And when we go through the different fish groups, actually the pattern remains the same. So no evidence of any negative effect of lionfish, or at least no evidence of a change or a decrease in abundance of any of these critical fish groups that we have on our reefs. But yet, for lionfish, you can see here from the pre-invasion to the post-invasion, we were starting to see them in many of the transects. So lionfish were definitely present at our sites. So we were surprised, because the idea is that we were going to see a decrease because of the presence of this voracious predator. But let's dig into the data a little bit deeper. And what is it we found? Well, we found that overall, even though lionfish were found at our sites, their abundance was generally low. So the highest, about 1.2 individuals per 100 meters square. When you look at the regional levels, if you look across the region in terms of what we can find in other locations, we're actually on the lower end in terms of lionfish abundance at any given site. And the other thing that we found, if you pay attention, we have depth here on the x-axis and abundance on the y-axis. You can see that as depth increases, the abundance of lionfish also increases. Lionfish tend to be much more abundant on the deeper reefs than on the shallower reefs. And just remember this point, right? Well, these 2 points, that overall abundance was low, and then second of all, they tended to be more abundant on the deeper reefs, as this should connect with some of the additional findings that I'll present in a minute or two. So now what happens to fisher landings, or fishers affected by the presence of lionfish? Was there any evidence of a change in the amount of fish that they were catching and in species composition? Well, what we found basically is that when we look at the spear fishers and we look at the trap fishers, if we look at the size, at the height of the bars, which in this particular case, showing fishing yields for spear fishers, this is a pre-invasion period and the post-invasion period, there's basically no difference. Very similar levels of catches. So no change in how much fishers are catching. That's for spear fishers. And the same applies to trap fishers. The amount of fish that were being caught before lionfish was established compared to the amount that was being caught on average. After the lionfish became established, there was no difference, basically. However, if you look in a little bit more detail in terms of the catch composition, and I'm going to focus specifically on the catch composition of spear fishers. This is the catch composition pre-invasion levels before the lionfish was established. You can see that half of the catch is actually made out of parrotfish. And if you know, parrotfish are important species not only economically because they get fish and we eat them, but also ecologically because they help keep the corals in a healthy state, coral reefs in a healthy state by feeding on macroalgae. So what do we find during the post-invasion period? Well, we found that lionfish had become the second most important item being fished, or fish group being fished by spear fishers, to the point where even the abundance of parrotfish had decreased marginally compared to what it used to catch in 2012. So if we do the math, we're catching the same amount of fish overall. But now in the post-invasion period, we're actually catching lionfish, which we're not catching in the pre-invasion period. What does that mean? Well, it suggests that there's a release in fishing pressure for the other fish groups because of the presence of lionfish. So to some degree, it suggests that we may actually have a positive outcome, after all, of the presence of lionfish by providing fishers with a new resource, and therefore releasing some of the pressure on the other fish groups. So we also, in addition to count fish, whether from the fisher catch or from transects underwater. We also did some surveys, and in fact, Robert, who is here, led the efforts here. And we asked dive operators during the COVID period to rank the sites on the basis of which sites, dive sites, are most visited and the dive sites that are least visited. And we know that all dive operators, to some degree, are engaged in culling lionfish or in spearfishing lionfish. Right? And we also asked them to tell us what was the general perception about the abundance of lionfish at those same sites. So we did that independently for each dive operator, and then we pulled the data. So what happens when we pull the data? What we found basically is that from the data that was provided to us by the dive operators, we find that lionfish abundance, and you can see this relationship, where lionfish abundance on the y-axis and number of dives per week on the x-axis, there's actually a negative relationship, whereby sites that are visited very rarely tend to have, on average, higher abundance of lionfish than sites that are visited regularly. What does this suggest? Well, it suggests that recreational spearfishing seems to also be having an effect in maintaining lionfish abundance at low levels. And this is an image of a management technique that seems to be very effective at maintaining those low levels of lionfish abundance. And this also aligns, if you remember, I mentioned that there was a relationship between lionfish abundance and depth. We know that usually spearfishing, it's the deeper you go when you spearfish, the less time you are going to be able to stay underwater, right? So the higher abundance of lionfish on deeper reefs probably is the result of lower levels of spearfishing compared to shallower reefs. So just to summarize, lionfish arrived to Barbados in 2012. Lionfish abundance is generally low, especially for shallow reefs, which is the ones that we surveyed. We found no evidence of negative effects of lionfish. So this doom and gloom picture that had been sort of that we expected to see never happened. In fact, actually, what we found is that spearfishers appear to be benefiting from the presence of lionfish. And to some degree, some of the fish groups that have been traditionally been fished by spearfishers may also be benefiting by the presence of lionfish by having lower fishing pressure on themselves. And therefore, from a management perspective, the recommendation should be that we should continue promoting the spearfishing, both recreationally and commercially, of lionfish to ensure that we maintain their numbers down. So that's the first story I wanted to share with you. The second story is also about a marine organism that has nothing to do with lionfish. And this is an interesting one. And Robert, who is here again, is the one who's been leading this. So some corals are also interestingly, very popular in the aquarium trade. And they're beautiful, large calyx or large polyp corals, as you can see here, with tentacles that extend quite far, big tentacles, yellowish, orangish. And when I do my teaching of coral ecology, the typical example of showing how a coral fits, you usually use one of these guys because they're so easy to use as model systems. Anyway, they're not from the Caribbean, right? They belong in the Indo-Pacific. And it turns out that we actually do have them here in Barbados. And we started to realize that we actually do have them in Barbados very recently, even though they probably have been here for much longer periods than we think. So they're native to the Indo-Pacific, first reported in the Caribbean in the early 1950s. And in Barbados, when we started looking into them, that was 2018. But then we've looked at pictures of dive sites, especially at night, and we were able to trace at least the presence of sun corals down to 2010. It is likely that they were here even before that, but nobody really noticed. Unlike many other corals, they do not engage in a symbiotic relationship with zooxanthellae. So they do not require light. And because of that, they do not also contribute to calcification in any substantial way. They do not contribute to reef formation. So they are different from any other stony corals in that regard. And in fact, you can often find them in shaded areas, which is not typically what you would expect from a coral. They have amazing capacity to reproduce and regenerate. And interesting enough, they tend to be found mostly on artificial structures. Now, in Brazil, where they can also be found, they've actually been identified as invasive in the sense that they can actually disrupt the native benthic communities of organisms. They can displace an endemic coral species over there, and they've been shown also to change, to have a strong impact on the community of organisms that typically you would find on rocky reefs in Brazil. And obviously that is cause for concern. So we actually also looked into we wanted to get a better sense of where we find these corals here in Barbados. And also we wanted to get a sense of, if we decide to remove them, if the recommendation is to remove them before they become overly abundant, how long would it take for them to come back? Because once they arrive to a location, it's very difficult to completely eliminate. You can control sometimes, but completely eradicate may be difficult. So Robert spent how many months? You don't have to say. He basically made a list of all the artificial structures that we have on the west and south coast of Barbados and went to nearly all of them with a flashlight at night, often because it's easier to see them at night when they have the polyps extended. And basically identify all the structures where you could actually find the coral. And that's him in there. And those are the locations of the site that he visited while he was doing this research. What did he find? Well, he found that 60% approximately of all artificial structures already had some corals on them. So these guys were already well established along the south and west coast of Barbados. Interestingly enough, he also found that structures that were made out of metal were more likely to have some corals than structures that were not made out of metal. So what do we mean by a structure made out of metal? Well, a shipwreck or a metal mooring. Those types of structures appear to have more frequently some corals than those structures that did not have metals, like breakwaters, for instance. We are still trying to understand why that is the case. If you're going to have a question about that, we don't really know. So I'm just preempting your questions. The other thing that we actually did is, okay, if we wanted to remove these guys, what is the best way to do that? So in the literature, in Brazil, they're using just scrape it. They're just scraping the corals off artificial structures. But this coral is unusual in that it has very high regeneration capacity. If you leave a little bit of tissue, it may actually be able to regenerate from that and come back in very rapid time when we think about how long corals typically require to grow. These are unusually fast-growing species. So what we did here in the Trident, which is one of the deeper shipwrecks in Coral Bay, which is at the intermediate levels of colonization of some corals, we established quadrats where we applied a complete removal treatment, where we removed all some corals, and then we left others as controls. And then we monitor this over time to see what happened. In other words, to get a sense of how long it would take for the corals to come back, for some corals to come back. And I'm just showing here the data, basically. So this is where we started the experiment. So we had quadrats where we had about 35 coral polyps per quadrat. Then some of the quadrats were completely scraped off, some corals, and left untouched after that, whereas we left the other ones as controls. And you can see here that there was a big drop, obviously, because we had just scraped everything off. After a month, we had a little bit of a recovery, and then it remained relatively stable afterwards. And we've kept monitoring this up until now, and it hasn't recovered, basically. It has remained relatively low compared to the controls, meaning that scraping will be a very effective way of controlling the abundance of corals wherever we see them, if we decide that we want to go that way, where they are right now. So just to summarize this part, which is the last part, some corals in Barbados, at least since 2010, it is likely that they were here before that. And if you have any pictures that go back even further where you have a sun coral, please let us know, because it would be nice to know when they were first spotted here. Now, they're found mostly in submerged artificial structures, and this is the tricky one, because they're not found on natural reefs. They seem to be constrained to artificial structures. So they're not going to interact with normal corals, with the reef-building corals. They're going to be interacting with the types of organisms that live on artificial structures, particularly on the shaded areas of artificial structures. It's a very specific type of community made out of sponges predominantly. Can reach high abundances in some structures. So if you go to the cement factory here, it's a beautiful dive, because especially at night, because they all extend the tentacles. I mean, it is really a nice thing to see, even though it's still an invasive. And if you wanted to remove them, it seems like an effective way. Simply scraping off with a knife or any similar type of device would probably work and keep their abundances down. But all this to say that we need to maintain monitoring. We need to ensure that there is not a jump from artificial structures to natural reefs at this stage. So monitoring should be ongoing. And if at some point we decide that it's time to remove them, at least we know how to go about doing it. And with this, I would like to bring the talk to a close and say thank you for coming. And obviously, we are happy to take any questions you may have.
So thank you, Julie, Darren, and Henri. Right, so I'll allow any questions from the audience. Everyone understood everything? So it's time for the quiz. Yes? Right. So I'll go first. So this is for Professor Horrocks. I think I missed it in your talk with respect to Redondo Island, right? So what did they do with the invasive species that were there? Oh.
Well, with Redondo, it's a small island. They poisoned the rats, and they were meticulous in the way in which they poisoned them. They, every single square meter of the island, they set poison, and basically kept poisoning and poisoning until no more rats were seen. And then with the goats, they took them off the island, and the goats are doing very well in Antigua, because what's interesting about them is that they survived on that island where there's really no standing water at all. It's just like a rocky outcrop. And they seem to have some kind of tolerance for drinking seawater. And so it was considered that they might become a very valuable resource, with climate change and salinization of water bodies, that maybe this is a species of goat that might have some value in the future. So the goats are doing very well and thriving and very happy on Antigua. The rats, not so happy.
No questions yet? So, okay, excellent. I just wanted to say, I wonder if, do they eat the goats? Do they eat goats in Antigua? Yes? Yes, excellent. So that's a new food source, because goat meat tends to be fairly expensive. Anyway, so that's a good food source. All right, you have questions? You want to use the mic? Yes. To your left there.
Hi, good evening. Good evening. My name is Hallam. Hope I have one silly question and then another one. The silly one is to Henri. Maybe went over my head, but my understanding is that the invasive species of coral doesn't pose a problem to Barbados, but you're basically scraping them off and removing them and that sort of thing because they're more focused on metallic structures such as sunken ships rather than coral reefs. Did I get that correct, or did it go on my head?
No, you're right. Right now, they seem to be confined to artificial structures, not to natural reefs in Barbados.
So it's not a problem then for our coral reefs, is that what you're saying?
I'm not saying that. I'm saying that they're confined to artificial structures and that we should keep on monitoring.
Right.
Now, bear in mind that, for instance, in Brazil, where they have become a nuisance, they have become a nuisance because they managed to jump from artificial structures to natural reefs. However, Brazil is a very different environment from ours. So what happens over there may not necessarily translate in terms of what happens over here. So what I would say is that right now we're monitoring it, and we haven't seen any sun coral jump onto a natural reef, at least not in any significant numbers. But that's not to say that it may not happen.
So the jury is out then?
So,
The jury is out then in terms of...
yeah, no, I think we need to keep on monitoring.
So my
like most
question...
invasives, yes.
Okay, so my question to Dr. Browne has to do with, I'm gathering that there is a potentially serious problem with this invasive species of worms, yeah, flatworms, yeah. My question is, to what extent are farmers aware of the problem, and what are these communities doing about it? Thank you.
Right, yes, in response to that question, we are in contact with the Ministry of Agriculture, so they're aware that the worm is here, they're aware that it's spreading. We have been getting information, we've tried to involve farmers, in part the Barbados, the Organic Growers Association is on board. And more likely than not, the Organic Growers Association are the ones that are going to be more heavily impacted by the worm, because the New Guinea flatworms are sensitive to the same things that earthworms are. So if you use broad spectrum pesticides and things like that, they're going to be affected. The thing about these worms is that they can simply burrow a few, 30, 40 centimeters into the soil, rest there and wait until the surface is clear and safe for them to come back out again. So farmers are aware, the Ministry of Agriculture is aware, but in terms of control, the only way to do broad control of these worms would be to essentially endanger some of the same species that we're worried about them eradicating. Because the same things that are going to be affecting the New Guinea flatworm are going to affect earthworms. All right, so the only way to do a sort of broad spectrum control would be to find some sort of pesticide that works on these worms, but not on earthworms, which is something that experimentation could potentially yield, but we're not currently there yet. What I can say is that for the residential properties, what we have been recommending is that persons use vinegar or salt to deal with the worm. So the thing about flatworms is that they can flatten themselves to just a few millimeters thick, and so they can get through all kinds of little spaces. So one of the problems that the residential property owners were complaining about was that these worms, they'll get through windows, they'll get under doors, they'll end up in the kitchen, they'll end up in the bathroom, they're coming into the house. And so if they treated them with vinegar, they actually, they almost dissolve into liquid. Treating with salt, same thing. It's not something that you can really do in a garden, and it's not something that you can do in a field, but at least for a residence, that's one means of control. But in terms of controlling these worms, the best approach is really to keep them from spreading, to limit the spread. Because once they're present, it's really, really difficult to get rid of them. And so one of the things that we're starting to talk about is making sure that soil is treated before it moves from one location in Barbados to another location. Right? So that you can actually potentially get rid of any worms, or at least check for egg cocoons and things like that. Once again, that's another thing that it can be very difficult, because if the worm is present in a field, a tractor drives through that field, crushes a worm, and a piece of that worm ends up in the soil on the tire of the tractor, it drives to another farm, you've inoculated that farm with the worm. So it's very difficult, but right now the best approach is, for persons, who see worms at their residence, treat them individually, and we can focus then on trying to limit the spread of the worms in soil.
Do you have any other questions for Dr. Browne before he sits?
Yes.
Want to use the mic? The mic. This side and then.
Good evening. My question relates to the same thing. I was thinking in terms of, how the giant African snails spread through the same transport of topsoil when people were clearing, and a lot of people clear their land in preparation for building, and then all of a sudden it ends up from St. George into St. Michael into St. James because people are selling topsoil. And I was wondering if that was another mechanism by which it could be being spread, and whether or not, when you had done your investigations and found those clusters in Christchurch, if it was related to the same sort of like topsoil areas of accumulation that were then being sold to different places, etc.
Yes, so that was something that we suspected, especially when we look at how the spread has happened. So you have these hot spots in Christchurch, and then just one point here in St. Philip and one point in St. James. The unfortunate thing is that when we were dealing with property owners, nobody could tell us where they gotten their topsoil from. So we suspect that that's what happened. But because, someone would say, oh, there was this guy, he provides topsoil to different people and so on. So there was no way to maybe get a sample or find out where the topsoil had come from, so that we can go to that site and confirm and maybe find a new location. Or maybe some of this topsoil was coming from Christchurch, near the hotspots. We're not sure. So I think the goal there is to just get more information, keep research, and try and find out if some of these points that we see, these locations, are actually near to areas where topsoil is generally being removed and distributed, and confirm that. But right now, we're not sure.
So my concern is along the same lines. Two things. You mentioned the use of vinegar and salt.
Yes.
And I know sometimes salt causes them to eject their pods. So salt may actually, if you put salt on them, you actually cause them to reproduce faster.
Okay. So no. So usually what happens? Yes, I would generally recommend vinegar over salt, in part because the salt then leaches. If you're doing it on soil, the salt leaches out. But the worms that are carrying a cocoon, it's actually fairly obvious. And what happens is not that it's in the case of the unique flatworm, not so much that they eject the cocoon when they're exposed to salt, but how do I describe this? So these worms, and I'll just brace yourselves. The flatworms are weird, okay? So there's an anterior end, there's a posterior end. The anterior end has eyes. The mouth is not at the anterior end. The mouth is about a third to half of the way down the body, and the mouth is also the anus. All right? And so what happens when you expose them to vinegar or salt is that the thin space above where that mouth is the skin begins to dissolve, and usually what happens is that the pharynx or the mouth just pops out through that area. All right? But generally, so yes, like I said, worms are weird. Yes, so it's not necessarily that it causes them to reproduce more quickly. And if the egg cocoon actually comes out, then it can be destroyed. But what's actually more dangerous is what people do as their first instinct, which is crush them. So the first instinct is not to treat them with anything. It's to crush the worm. But remember,
Each piece
becomes a new worm, right?
Yes.
So that's the real danger there. But yes, if any parts of the worm, the vinegar actually does work on the egg cocoons as well.
Lime juice as well.
Yes.
Right. The other thing is, have you thought about traps? The same way we would trap giant African snails, we have worm traps that may not be as specific to this particular worm, but at least it is going to generate some attraction, even if you use a pheromone for this particular pest.
A pheromone, that would be pretty interesting. That's something that we could potentially look into. We have tried traps. We had one, I'm very, very grateful to this person. One person at a property, she was willing to let us, for over the course of about 2 months, put flat bits of perspex and try to essentially capture moisture underneath them. At the time, we were finding the worms mostly underneath flower pots and things like that. So we were kind of trying to recreate that environment in her yard and see if the worms would be attracted to that. And what we found was that it's, unless you use a really heavy material, right, it doesn't trap enough moisture to attract the worm. And then they're free to move from underneath it, especially at night because they're hunting. So they're looking for other organisms which may not necessarily be attracted to as moist an area as they are. So we did try traps. I think we were successful in attracting just 1 or 2 worms to the traps that we laid down. And then we continued to find the worms, initially, as I said, underneath flower pots. But this was end of 2020. I don't know if anybody remembers how wet that was. But yeah, by the time we'd been searching for about 3 weeks, the worms, it was as though they were emboldened. They were everywhere. They were out in the open, and we thought that they would be retreating to the traps, but they weren't. So they weren't retreating to the traps. They weren't found underneath the traps during the daytime, but so they can burrow in the soil. So there's a decent chance that they'll just burrow into the soil and hide there, and then come out at night as well.
One suggestion would be to use bear. I know it's a good waste of good bear, but they're pretty attracted to bear as well.
Okay, so that's something that we can try. The other question that you asked about pheromones. So each worm, once again, worms are weird. Each worm is both male and female. All right. So there's a chance that we can maybe isolate a pheromone, but as they crawl along, they're leaving a trail of mucus, and they may be using that trail of mucus to find a potential mate and that sort of thing. So if we may be able to collect some of the mucus and see if there's something that we can isolate that acts as an attractant for other worms. But we did have Connor Blades, who Professor Horrocks mentioned earlier. He has been doing he did some work with capturing insects with bear. So that's something that I can discuss with him and see if we can give it a try, especially at one of these sites where we have quite a large population of worms.
And I tend to mix the bear with a little bit of kitchen scrap, so cut cabbage, lettuce, whatever. The other thing is, you mentioned that the Ministry of Agriculture is aware of this invasive.
Yeah.
Are you aware of any information packs that have been prepared to alert farmers to be on the lookout? What should they look for? Is there any type of frass that they should be looking for? Is it a notifiable pest as yet? How should they prevent it from coming on their farms, and treatment on farms? Because it could seriously damage your native earthworm population.
This is true. There is no information package as yet, but this is something that we are working on as we move along. One of the things is that this is really an emergent threat, so we're still kind of working out exactly how they're spreading. We suspect that it's actually exchange of soil.
More than likely,
more likely that's what's happening. We're not absolutely sure. So there are a few things that we need to verify first. But yes, that's something that we are looking forward to doing.
With so many species migrating in response to climate change, are we developing ways to anticipate new ecosystem structure and function, and perhaps being able to understand whether an invasive species might down the road become a healthy part of that new structure and function?
It's a very good question. Yeah, so I think in some cases, what we actually tend to do is we tend to monitor invasive species first. So once an invasion occurs, we need data. So as with the case of the sun coral with Dr. Vallès, the species is present. It may have the potential to become a problem, but we're not sure. So we keep monitoring it, but we make sure that the public is aware. We make sure the person's aware that it's present. In some cases, the technique that I use, the species distribution model, the habitat suitability model, is something that can be used to predict a suitable habitat for a species. So we can use that if, for example, we have points where a species is popping up around the Caribbean, and we have those environmental variables, we can potentially extrapolate and see and make a educated guess as to where they may turn up next, right? So we can potentially predict the introduction of an invasive species. But in terms of whether or not that species is going to become, just a natural part of the environment, and it's going to be an individual situation, a case-by-case situation. For example, in the case of the invasive planarians, there's a chance that they will help in terms of controlling giant African snails, in terms of controlling snails that are going to be attacking crops and things like that. And they may not be as big a problem when it comes to potentially feeding our earthworms, but we're not sure. All right. So there's always the potential for an invasive species or an alien species to have negative impacts. There's a potential for it to have somewhat positive impacts, but we really don't know for sure until they're actually established in the range and we can actually see what the interactions are like.
This is a general question as it relates to the perspectives of people when considering what species are invasive and what aren't. So you have your technical definition, but then where humans are concerned, you have the novel, exciting, good-looking species, or the mammals that are not a big concern because they're not like the collared dove, for example. People would not necessarily consider that invasive because it's just another bird. So your flatworms, which are disgusting and melting and pooping and all of those things, people are like, oh my God, kill them all. But then you have something like the sun coral that the divers are like, well, climate change, poor water quality, reefs are dying, but here we have these really beautiful coral that we can take people and show them, which is a benefit to tourism, and it's going to perhaps negatively impact the reef health in general, but produce some dollars in the short term. Or maybe they're thinking the coral is going to die anyway, so at least we have this. How do you really change the perspectives of people when it comes to the eradication of invasive species and overcome that bias where things like monkeys that are damaging crops are seen as Lucifer, but things that are a little bit more pretty or a little more innocent seeming are readily accepted, especially in a country like Barbados where our general biodiversity is quite poor?
Okay, once again, another good question. I think in that sort of situation, the focus has to be on educating everyone on the wider impacts of the invasive species. So there may be the aesthetic aspects of it. So it's pretty to look at. It improves your dive and so on. But this is the history of this particular species. This is where it has been introduced before. This has been the impact when it was introduced before. For a certain period of time, it was benign, and then after a certain amount of time, this is what happened, and this was the consequence. And so, if we want to preserve what our current biodiversity, we need to act now, as opposed to looking at the species and saying, oh, well, I really want to look at it, so, and it's earning me money right now, so we should keep it around.
You will get to afterwards to say something.
I was just going to say that this is one of the problems with dealing with invasive species that are attractive or charismatic, and it's one of the problems that Barbados is facing with addressing the green monkey problem, because there is a fairly large segment of society who are not directly economically impacted by monkeys, who they don't see the monkeys as in the same way as farmers obviously do, who are experiencing a lot of crop damage. And these kinds of considerations, it's not just here, but when you have beautiful mute swans, for example, and people say that they have to control populations of beautiful birds, that causes a public outcry. It also happens with trees. Casuarina was one of those species which was shown on the cover slide. And, it's so much a part of the Barbadian landscape that we don't even really appreciate that it is, it has negative impacts on other plants. You would probably face a public outcry if you were to just cut down casuarina trees, certainly unless you were going to replace them with something else. But, we've become accustomed to them. And as Carla was saying, this country is full of introduced species. That, there are many people who think mongooses are native to the island. There are even people who think that the monkeys here are native to the island, don't realise that they've been introduced. We're so accustomed to seeing them, and we don't have a lot of biodiversity to look at other than them. So, it raises all kinds of ethical questions when it comes to control.
Thank you. That was actually one of my questions that I had here with respect. When does an invasive species stop being invasive?
It's a very good question.
Yeah. Any other questions from the audience? Nothing? Okay. So I have this is not, Henri and I can discuss this afterwards, but I'm going to throw the question out to him anyway, with respect to the sun coral and it being more prevalent on the metal parts of the wrecks and stuff like that. Have we done any testing to determine the metallic content of the sun corals versus the ones that are not on the metal, because they may be feeding off the metal?
Actually, I have a theory about why we found that they appear to be more likely to be found on metal structures. And my instinct is that most of the shipwrecks were being found in Coral Bay. So I think that what's happening is that sun corals appear to be more abundant at Coral Bay than other places. So it's not really so much the metal structure, more so the fact that the shipwrecks are actually in a place where there's already quite a bit of sun coral. So that's something that we need to look into more detail, because I suspect it's simply an artifact of the fact that the shipwrecks appear to be at a location that seems to be quite abundant in terms of sun corals, not necessarily the metal structure itself. So it's a little bit of an artifact perhaps from the study, and we will need to do some kind of experiment to actually really tease out what exactly metal structure is the location. So there's nothing in the literature that suggests that metals would attract sun corals more than other types of structures. So I suspect it may be simply the fact that it happens that Coral Bay has quite a bit of shipwrecks.
Good evening, Henri, landfish. Well, when we first heard about the landfish coming, we thought they would just decimate the reef. So it's really great to hear they haven't done that. But two questions: what exactly do they eat, or do they really eat everything on the reef? And how, like juveniles of everything. And the other question is, has there been any impact on, say, something like lobsters?
I don't know about the lobsters, to be honest.
But they eat everything.
Yeah, they're opportunistic feeders. They will eat on whatever is most available. So for our reefs, that's damselfish and gobies and things like that, which are very abundant. And in fact, when you flesh out the data a little bit more, we had a presentation a couple of weeks ago at another conference where we actually showed that some evidence supporting that sites that have lionfish have fewer damselfish. But the damselfish are one of those species that has been released from predation. Right? They're everywhere. Fish that used to eat them are no longer here. So the lionfish may actually be reduced in their abundance in a way that used to be in the past. Right? And damselfish, too many damselfish is probably not good for the reef either, because they create these algal gardens. They can take the space of other benthic organisms through their algal gardening activities. These are species that are going to create a little garden of turf and then sort of nurture it and maintain it. So when you have too many of them, the whole reef becomes a big garden. Right? So that may be an interesting story that to explore further. But they're opportunistic feeders that will feed on whatever moves. In terms of lobsters, the best way to look into that is looking at their stomachs. When you catch them, figure out what they're actually. But we didn't see. I didn't see any lobster during our surveys. I think those two may not be as abundant as they used to be. So I don't recall having seen any lobsters. So. Yep. Okay.
So according to my watch, it is about 12 past 8? No? Yes, y'all have me worried there for a minute. So one of the things I learned from the lecture today, beside the invasive species, etc., is that we do have a potential business in producing pictures of the sun coral as well as the lionfish, which I think are pretty, yes, and selling it. We had a whole hotel in St. Philip that was built, right? The window? Wouldn't it have been nice to have these portraits of these species that are endemic, not really, to Barbados, yes, on the walls. So, ladies and gentlemen, because we do not have any more questions, I would like to bring this lecture to a close. But of course, first off, I have to give thanks to all the persons who contributed to the success of the lecture. So first off, the presenters, Professor Julia Horrocks, Dr. Henri Vallès, and Dr. Darren Browne, for essentially making us aware of species in Barbados that we see every day that we probably think really belong to Barbados, and they're not. So thanks for making us aware of that. Knowledge is always a good thing. To Dr. Shelley-Ann Cox in the Division of Fisheries for allowing us to have the public lecture in this room. To the staff of the Department of Biological and Chemical Sciences for all the behind-the-scenes planning, especially to Ms. Ariane Franklin. The staff of the Faculty Office in the Faculty of Science and Technology at the UWI. The Director and staff of the Office of Marketing and Communications, Public Information, inclusive of Ms. Carol Williams. The press that is present with us today, and the audience, of course, because without the audience, we would not have had this lecture, because we can always stay up on the hill and speak amongst ourselves. Yes, but I hope that today's lecture has inspired some of you all, awakened interest in some of you all, and please know that we on the Hill are always open to having discussions with members of the public at any point in time. So, and we are always willing to share our knowledge, and there's a lot of knowledge that you all don't know about. So I expect to see more of you all visiting the Cave Hill campus and interacting with us up on the Hill. So thank you all very much, and please get home safely.